And today we are hosting today's seminar together with the Southern African Society for Systematic Biology. Before I am handing it over to Fanos to introduce SASSB to us, I would like to thank to all of you for all of our speakers for their willingness to join and give their presentation, and for all of you for joining. Please feel free to type your questions in the chat box during the talks. And Fanos, it's over to you.
Thank you very much, Neriman. On behalf of the Southern Africa Society for Systematic Biology, I also would like to welcome you to our first day seminar. And it's just very fortunate that we could combine the two today and sort of get the fields together. So FABI is very pleased that we can also join up with the society. The society's focus is on systematics and covers all the sort of biological fields. So you have microbiologists, you have mammal taxonomists, etc. So it's quite a wide range, but we share a passion for systematics, and therefore we're also very excited that you join us today in this theme. The outlay of it will be a little bit different from the normal FABI international seminars, in the sense that typically for the FABI seminars, we only have the international speaker and then a question and answer session. But because of we, as the SASSB, we are a society that would like to promote also our young researchers, we have opted for a webinar where we have an international speaker and then two local speakers, typically students. And today we will have two students joining us. Wilma Nel and Aiden Visagie will give a short presentation afterwards. So it will be a slightly longer than usual, but I hope that you can stay until the end and enjoy it with us. And with that, I hand over to Brenda to introduce David to us.
Thank you very much, Fanos. Good afternoon or almost good evening to everybody. I'm not quite too sure. Well, we certainly know that we've got at least one person in the audience who's logged in from South America. So South Americans are pretty much in the same time zone as our speaker, David Hibbett. So good morning to those of you who are in morning time. And David, I hope you've got a nice cup of coffee to wake you up properly there. So it gives me a huge amount of pleasure to introduce David Hibbett to you. David is a very, very good friend, personal friend, and a friend of FABI. David agreed many years ago, we were actually working out, it was 8 years ago, to come out and do a PhD oral defense for Tuan Dong, who is actually in this audience today. So David, we have very, very fond memories of you being out here with us, and we look forward to your next visit. So David is a professor in the Department of Biology at Clark University in Massachusetts. And David has in actual fact been at Clark University for, are we allowed to say, a very long time, David? Before coming to Clark, he completed his PhD with another very well-known mycologist and good friend of ours, Rita Spilgeles. And David has been the recipient of many awards. And I think in the context of today, I want to focus on awards from the Morphological Society of America. This is a society that certainly I have attended many meetings of. It's, I think, probably the nicest mycological society in the world, but I don't know if this has been recorded, maybe that's not a good thing to say. So David, many years ago, got the Alexopoulos Award, and David is, of course, now a fellow of the MSA and past president. So David, we did invite members of the MSA to attend today, and I'm not quite too sure if we have some MSA members who've joined, and certainly we welcome them. And David is also on the highly cited researcher list, and we kind of look at this list, and certainly as mycologists, somehow we don't get the citations that some of the medical people or some of the biochemists do. So I think that speaks hugely to the the international profile that David's research is getting. And David, I actually asked David to send me his CV to be able to introduce him, and I'm not going to focus too much on it. Yeah, he's done all the things that he's meant to have done, raised lots of money, probably spent a hell of a lot of time writing grants to be able to do that, and has a very impressive list of publications. And I invite you to go onto David's website. I actually did go onto the website, David. I rather like that blogger that you've got going there. That looks like fun. And have a look at what David's been up to. But I'll stop there, David. Thank you so much for being prepared to join us, and we look forward to your presentation.
Great. Thank you, Brenda, very much for that introduction. Thank you, everybody who was involved in inviting me to come here. It's a great pleasure and an honor, really, to be with you today. This is my first interaction with the SASSB, and I'm really pleased for that, to meet a new society. And of course, I'm very happy to be told I'm a friend of FABI. I feel very friendly towards FABI. My visit there years ago remains one of my most cherished, work-related visits, because it was not all work, and it was really quite wonderful, and I'm forever grateful for the hospitality that I received. So with that, I'm going to share my screen, and we'll start here today. And I'm going to talk to you about one of my favorite organisms. This is the genus Lentinula, which I haven't been giving talks on this very much in recent years. Mostly it's been phylogenomics related to wood decay and classification and things like that. But in recent years, I've come back to working on Lentinula, which is the Shiitake genus. And I'm going to tell you about some very old work, some not so old work, and some stuff that is still, frankly, quite raw and in progress. So I'm going to divide the talk into, let's see. Whoops, there we go. Into really 4 sections. First, I have to tell you about just give you some general background on Lentinula, tell you about the taxonomy of the group. I'm going to present some research that is in review right now on phylogenetic diversity in the group based on ITS and TEF1. Mostly what I'll talk about, I think, is phylogenomics and population genomics, and then I'll conclude with some outstanding mysteries, because even though this is a well-known group of fungi, I hope to convince you that there's still an awful lot to learn, a lot of discoveries probably to be made. Okay, so you all know this fungus. This is Lentinula edodes. This is the Shiitake mushroom in Japanese. That means oak mushroom. It's pyogo in Korean, and it's shangu in Chinese. And this is one of the most widely cultivated mushrooms in the world. There was a 2017 study, I've got a figure here, which suggested that it was the #1 mushroom in the world, but it's always flirting between Lentinula, Agaricus, and Pleurotus. But Lentinula is certainly one of the big ones. So everybody knows Shiitake mushrooms, very important commercially and of interest for potential medicinal properties, and so on. It was described in 1877 by Berkeley, the British mycologist M.J. Berkeley, as Agaricus edodes, because back then everything with gills was in Agaricus. Berkeley described it based on material that he'd received from the Challenger expedition, some material that was purchased in a shop in Japan. So it would have been a commercial sample of Shiitake that he described the species based on. There was some taxonomic controversy involving this extremely well-known organism. The great mycologist Rolf Singer, who was tremendously influential mycologist of the 20th century, put the species in the genus Lentinus. Another somewhat less famous, but also very influential mycologist, David Pegler, who actually did a monograph on both Lentinus and Lentinula, said, no, it's not a Lentinus. It's in the genus Lentinula, which he placed in what he was calling the tribe Calibii in the Agaricales, so white-spored agarics. So this was a big controversy for much of, I'd say, the late 20th century. Is Shiitake a Lentinus or is it a Lentinula? And I did my PhD a very long time ago on the genus Lentinus, and this is actually a figure from my PhD thesis. using what is now an embarrassingly small amount of data. Nonetheless, I think this holds up pretty well. We have a diverse set of mushrooms in this tree. The groups labeled 1, 2, 3 are lentinus in the broad sense. Here's Shiitake Lentinula edodes, closely related to a colibia. Remember, Pegler put it in the colibii with pretty strong support. So we figured that nailed it. We've done our due diligence now. I've finished with Shiitake. I can move on and just focus on lentinus for the rest of my PhD. And that's what I did. And so I was prepared to leave Lentinula behind. But as it happens, I did a postdoc, my first postdoc at the Pretoria Mycological Institute, which is in Japan. They are funded in large part by the Shiitake industry. They excel at Shiitake cultivation and strain development and so on. They're intensely interested in Shiitake, so I found myself drawn back into the world of Lentinula. And this is me a very long time ago, looking much younger and rather happy. You'll notice the beers on the table here. This was a party with my colleagues. So with my friends from TMI, I launched a little molecular systematics project on Lentinula using ITS. And our goal was really to test Pegler's classification system. So this is, so we'll run through that, focusing first on the Asian, Australasian things that Pegler recognized, and then talk about the American things. So Pegler's classification was very strongly influenced not just by morphology, but also by geography. And he divided Lentinula into 5 species, 3 in Asia and Australasia, and then 2 in the Americas. So the things in Asia, Australasia included edodes, Shiitake, which you've now seen pictures of and which you knew already, something called Laguardia from Southeast Asia, so a tropical species, and then Novi Zelandii from New Zealand. And I'll show you a couple pictures of these species that you're probably not familiar with. Here are the sketches from Pegler's monograph, so edodes, Novi Zelandii, and Laguardia. Here's a photograph of Lentinula laguardia from Australia. It's got this kind of smooth, thin-fleshed pileus. And it's very tasty, by the way. I've collected this in New Guinea, and it's delicious. And then here's the New Zealand species, Lentinula Novi Zelandii, which looks really different. It's got this kind of fuzzy, hairy stipe and really heavy squamules on the cap and so on. And it's got this kind of dark aspect. So those were Pegler's concepts of the species in Asia, Australasia. We used ITS, very simple analysis, and those data suggested that there are actually 5 lineages in Asia and Australasia, shown here in this rather old tree from some publication of mine. So we gave these group names based on ITS clusters, and for the most part, we didn't do any taxonomy based on this work. We didn't translate this tree into formal taxonomy. So we had group 1, which is edodes, group 2, which is Laguardia from Australia and New Guinea, group 3 from New Zealand, group 4, which is another group from New Guinea, and I'll have more to say about this one in a minute, and then group 5 from China and Nepal. And I'll be referring to these ITS-based groups throughout the talk, so hopefully you'll get comfortable with that terminology. So here's a map just showing you where they all are. So group 1 in blue, so throughout continental Asia, Northeast Asia. Also, we had 1 isolate from Borneo, but also notice group 5, our 2 isolates of group 5 in China and in Nepal. Group 2 in New Guinea and in Australia and Tasmania. Group 4 also in New Guinea, very interesting. And then group 3 in New Zealand. Okay, so 5 ITS-based groups of Lentinula corresponding to 3 morphospecies in the sense of David Pegler. Now, my colleagues at TMI were very reluctant to translate those results into species descriptions, in part because almost all of the isolates throughout Asia, Australasia are still mating compatible. So based on that criterion, you could think of them as being all one species, even though using molecular data, we can see many independently evolving lineages. But it turns out not to be entirely clean on the mating front. So this is a figure from a paper by Shimomura and colleagues. I've taken their table and just sort of annotated it. So the colored boxes refer to different groups of Lentinula. So this is group 4 from New Guinea. This is group 2 from New Guinea. And these are groups 1, 3, and 5 from throughout Asia. And wherever you see a plus sign, that means that these things are mating compatible based on the criterion of being able to form clamp connections in culture. But what you can see here is that group 4 and group 2 are completely mating incompatible. So these sympatric populations in New Guinea are reproductively isolated. And the plus signs in parentheses on this yellow background indicate partial mating compatibility. So group 4 is partially or fully reproductively isolated from other things in Asia, Australasia. And on that basis, my colleagues at TMI said that these Shiitake populations in Australasia are in the process of speciation, but weren't quite ready to pull the trigger and actually describe new species. Okay, so that's Asia, Australasia. We're going to turn now to Pegler's concept of Lentinula in the Americas. And again, there was a pretty strong geographic component and morphological component as well. So there were 2 species that he recognized. Lentinula boreana in tropical America, he said, which covers a lot of territory. And then Lentinula huarapiensis, a very mysterious species known only from the type collection from Paraguay. So rather obscure species there. And here are the pictures from the monograph. So here's Lentinula boreana, and here's Lentinula huarapiensis described by Spegazzini. Now, the taxa in the Americas have been carved up into multiple species by Juan Mata and Ron Peterson and colleagues based on mating compatibility and also ITS phylogenies. So based on those criteria, Mata and Peterson distinguished 2 new species, Lentinula raphanica and Lentinula aciculospora from Lentinula boreana. And here are some pictures of those. So here's raphanica from Brazil. Here's a picture of Lentinula boreana taken in Mexico, and this is Lentinula aciculospora from Costa Rica. And these are all edible and have a characteristic kind of oniony or garlicky aroma, which is a trait shared by all Lentinula species. So this figure here kind of represents the paradigm based on ITS for much of the last 20 years, which suggests that there are 8 independent lineages in Lentinula that might warrant recognition as species, but not all of them have been formally described yet. And showing you the map with the distribution in the Americas, and in Asia and Australasia. So it's a fungus that is associated with the cuisines of Northeast Asia, but it really is very broadly distributed. Okay, next I want to bring you forward a little bit to a project that's currently in review. It's been submitted. We wanted to update our understanding of the phylogenetic diversity of Lentinula based on ITS, but also now bringing in a second marker, which is TEF1 alpha. And this has been a really big collaborative project headed largely by Nelson Manoli here, who's from Sao Paulo. This is Noemia Ishikawa, who's in Manaus, so a very strong Brazilian connection here. These are some folks from my lab, other collaborators whose pictures I'll show you later. Chao Chun is from Guangdong in China. Marisol was a postdoc in my lab who's now in Sweden. So it's been a really international research project, and that's been one of the things that I've found really fun about it. And these are Lentinula, of course. So what we did was we wanted to get a really broad sample of sequences from all over the world. And we got 343 ITS sequences together and 116 TF1 alphas, a bunch of new sequences, many mined from GenBank, mostly Edotes sorts of things. 60 sequences or 60 individuals came from some unassembled genomes of Lentinula Edotes in the broad sense that were published in 2016, from which we just extracted the ITS sequences. And I'll have more to say about those 60 unassembled Lentinula genomes in a little bit. So big picture, combining these data, we detect up to 13 groups, 5 in Asia, Australasia, so the same 5 I talked about before, and up to 8 in the Americas. So now we've got more resolution of lineages within the Americas. These are all candidate species and all pretty well supported by the traditional measures, bootstrapping and so on. So I'll focus first on the Asian Australasian side. So again, I told you there are these same 5 groups that we've been seeing all along. Group 1 from Borneo, lots of sequences from China, India, Japan, Korea, the Philippines, Thailand, the Russian Far East, and Vietnam. Group 2, again, New Guinea, Australia. Group 3, New Zealand, that's Lentinula novi-zelandii. Group 4, the second group from New Guinea. And then this group 5 group from China, India, Nepal, and Vietnam. Okay, so group 1 and group 5 overlapping in terms of their geographic range. So this is a simplified version of that tree I just showed you. This is the Asian Australasian group up here. And what you can see is that in this combined ITS-TEF1 data set, group 1 and group 5 are resolved as sister lineages, but with low bootstrap support for their monophyly. But if we look at those 2 genes individually, ITS on its own doesn't put group 1 and group 5 as sister taxa or in the same clade. So they're just different ITS types, not a lot of support for the deeper nodes in the tree. And TEF1 alone doesn't discriminate those individuals at all. So they're just, if you saw a phylogram of this, it would just be one big, completely unresolved comb with really no branch length within this group. So this made us scratch our heads and wonder if group 1 and group 5 were real. And this is the phylogenetic distribution, or excuse me, the geographic distribution of those groups. So group 1 and group 5 largely overlapping, and group 2 and group 4 sympatric in New Guinea, as you've seen before. Asia, Australasia, Lentinula raphanica, Lentinula boreana, and Lentinula aciculospora all contain multiple lineages. This boreana species complex may include actually 4 independently evolving lineages that we might recognize as species. And you can see the distributions listed here. And then again, here's just the geographic distribution of these putative species, ITS-TF1 based lineages, and some very provisional names for some of these in this figure here. Okay, so with more sampling, we're getting more resolution of multiple lineages, particularly in the Americas. Using the same data, we performed some molecular clock analyses, which suggest that this genus is roughly 50 million years old, give or take 10 or 15 million years on either side. So, take these with a grain of salt, but these are our approximate estimates of the age of Lentinula. We also looked at host range. So the word Shiitake in Japanese means oak mushroom because they grow on oaks. And all 13 of these independently evolving lineages are recorded or likely to be on Phagales, except for Lentinula aciculospora, and then this group 6 of Lentinula raphanica, which is on a very diverse group of hosts, all hardwoods, but not Phagales. So based on this tree and this mapping of hosts, we think it's likely that the ancestral host of Lentinula was Phagales, which is consistent with the age we get from the molecular clock analysis. Okay, so summing up that large phylogenetic analysis, we now think that there are about 13 lineages in Lentinula that might warrant recognition as species, with 5 in Asia, Australasia, and 8 in the Americas. So next we're going to turn to phylogenomics. So this phylogenetic work using ITS and TEF1 provided a really nice framework to design a sampling scheme for doing a genus level genome based analysis. So we've got a new set of collaborators, or an overlapping set of collaborators. The genome sequencing was all done by the Joint Genome Institute in California, which has been just a superb partner for my work for many years. Byung-Nam Min was the main postdoc on that. Carrie Berry, who insisted I use this silly picture. This looks like an organism you might have in South Africa. Carrie Berry was the project manager, and Igor Grigoriev is the head of the Fungal Genomes Initiative at the JGI. And then these other folks are collaborators and friends. I'll point out Kazuhisa Terashima from the Totori Mycological Institute, Ron Peterson, who I mentioned previously, and his former student Juan Mata. Chris Smith is in New Zealand, and others who have been really helpful with this work. So we generated 25 new genomes, which represent 11 of the 13 groups based on ITS and TEF1 from 10 different countries. And then we imported 4 genomes, 1 Novi Zelandi genome, and the rest all Edodes from China, Japan, and Korea. Okay, so we really think we got a pretty broad sample of diversity in Lentinula based on everything we know from ITS, TF1, and also biogeography, Pegler's monograph, and so on. So we obviously got all of Pegler's morphospecies represented in the data set. And if you want to play with the data, they're all available on the JGI Microcosm website, and we're frantically trying to write up a paper now on this work. And so all the work I'm going to present from this point on is not only unpublished, but we haven't finished the analyses yet. So this is very much work in progress, and I will really welcome your suggestions, your interpretations, your critical comments, because this work is not done. This is Miguel Naranjo Ortiz, who's a postdoc in my lab right now, and he's been doing pangenome analysis in Lentinula, which I'm not going to go into any detail. I'm showing you this slide mainly just to make the point that these Lentinula genomes are fairly typical for agaricomycetes, so on the order of 34 to 49 megabases with about 12 to 16,000 genes per genome. So fairly typical agaricomycete genomes. And Miguel has done a couple of sort of straight ahead phylogenomic analyses using OrthoFinder as the main tool for scoring the ortho groups from which the genes are pulled for doing phylogenetics. Dataset 1 includes a broad sample of outgroups from throughout the Agaricales. It upholds the placement of Lentinula within the Calibii sensu Pegler. So this is dataset 1, which is the broad analysis across Agaricales. Dataset 2 is more focused within Lentinula, and now I'll talk about what's in the tree. And basically, it's really consistent with prior taxonomy. So we've got Lentinula raphanica, Lentinula boreana, and then in the New World, Lentinula aciculospora from Costa Rica is up here, sister group to things from Asia, Australasia. So here's this group 4 from New Guinea. Here's just in this particular data set, there's just one isolate of Novi Zelandi from New Zealand, although we now have 2. Similarly, there's one isolate of Lateridia from Australia in group 2, but we have 2 of those now. And then 2 group 5 and 2 and 4 group 1 based on ITS. And in this analysis, they come out as monophyletic sister lineages, but that you'll see later is an artifact of low sampling. One of the surprising results from this analysis, of course, is that aciculospora from Costa Rica is not the sister group of the other things from the Americas as it was in the ITS trees. Rather, it's the sister group, albeit on a very deep node, to all the things in Asia, Australasia. And that's weird from a biogeographic perspective. So we tested that topology, used a bunch of different topology tests with dataset 1 and dataset 2. And the bottom line is that the topology that we get that puts aciculospora as a sister group of the Asian Australasian things is the only one that we can't reject. So we feel pretty confident that the closest relative of the Asian Shiitake or the Asian Shiitake group is actually something that lives in Costa Rica and Ecuador, which is surprising. And here is aciculospora, which in this photo at least looks an awful lot like Lentinula edodes. Okay, now Next, we wanted to focus more intensely on the Asian things, Lentinula idoti sensu lato, because of course we're interested in the origins of cultivated shiitake mushrooms. And for this purpose, we were really fortunate to have access to these 60 genomes that Yang Xiao from Huazhong University in Wuhan had published. And he had published this just as unassembled raw reads and had done some SNP analyses. This is a figure from his 2016 paper, so kind of a while ago. And we were able to, and Yang Xiao is now collaborating with us, for which we're quite grateful. But we, and when I say we, I mean Byung-Nam Min from JGI, was able to get his unassembled data and assemble them into genomes with pretty good completeness based on Busco scores. So we were able to assemble those and start analyzing them. And here's a phylogeny now of Lentinula that includes 77 genomes, 56 from the Xiao et al. Study, and 19 from JGI. So this doesn't have every single genome we now have available, and then a couple of published things. This topology is from a kind of quick and dirty concatenated single copy gene data set, 415 genes analyzed with FastTree. We've done a bunch of other analyses of these data now, including species tree analyses using a program called Astral. They all give us basically the same answer. This group up here, this big group here, this is Lentinula idotes in the broad sense. So this is the group within which shiitake mushrooms, the shiitake mushrooms of commerce, are nested. And it's a little bit of a complicated slide, so let me just explain what you're looking at here. So the codes on the tips of the tree in red correspond to cultivars, so samples from cultivated shiitake mushrooms. Those in blue are wild collected strains. And you can see that the cultivars and the wild things are kind of all mixed together in this group, which we call cultivars plus wild. This group down here is all wild strains. We call this the wild group. And then there's this group here that's got one cultivar recently cultivated and some wild things, which we call the mixed group. So that's the distribution of cultivated versus wild collected strains in Idoti sensu lato. Then these G1, G5 notations on the right indicate the ITS type, the dominant ITS type that we either amplified with PCR or extracted from the raw reads using sort of an in silico PCR type method. And what you can see is that group 1 and group 5 do not sort out as individual lineages. They're sort of all mixed together. And this wild group has many of these, has a bunch of G5 type ITSs scattered within it. So this is in conflict with the ITS based analysis. The next thing that we turn to, and this is, by the way, this is the first time I've ever shown this figure in a talk. So this is really brand new. These data actually, this is an analysis done by a postdoc in my lab, Brian Looney, and he gave me this figure last week. So I'm happy to share it with you now. This is an analysis using a program called STACY, which is a Bayesian species delimitation program that models incomplete lineage sorting, or it uses the coalescent, in other words. And this is based on 50 single copy genes used to generate this tree. And then the STACY program estimates what are the likely independent lineages, which may correspond to species, taking into account coalescence. And we're going to focus only on this big square down here, which is idotes in the broad sense, so Lentinula idoti sensu lato. So here is the wild group, which contains G1 and G5 type ITS. Here is the cultivars plus wild group, which is only G1. And this is the mixed group in the middle. So again, with only G1 ITS. So I'll just go back to that tree really quick so you can see there's wild, mixed, cultivars plus wild. And so here they are again, wild, mixed, and cultivars plus wild. So this analysis suggests that there are at least 2 independently evolving lineages in Lentinula idoti sensu lato, taking into account incomplete lineage sorting. Now, Stacey analyses are sensitive to a setting called the collapse height, which is basically has to do with the depth at which genes coalesce or are allowed to coalesce within a discrete lineage, if I'm explaining that properly. And what this figure shows you is a bunch of different Stacey runs at different collapse heights. So this would, down on the lower right, this is closest to the figure I just showed you, and it's most inclusive, right? So it's going to give you bigger groups, so allowing genes to coalesce deeper in the history of the species. On the upper left, it's a more restrictive collapse height, so it's going to start to parse out many more independent lineages. So it's kind of, there's an element of subjectivity to deciding at what point you have results that correspond to what you might consider species. Based on calibrating with respect to other, I would say, well-delimited species like Novi Zelandi, I'm most comfortable with this delimitation, which is why this is the one I focused on first. But what you can see is that the wild group has more genetic diversity than the cultivars group. The gene lineages coalesce deeper in the tree, whereas in the cultivars plus wild group, they coalesce more quickly, suggesting that there's just less genetic diversity. But this is all, so this is an interesting method that takes into account incomplete lineage sorting, but it's still very much doing a gene-based phylogenetic analysis, right? So we wanted to take a more sort of population level approach and look at SNPs. So look at single nucleotide polymorphisms within Edodes sensulato. So for that purpose, we used admixture to sample about 10,000 SNPs. And we estimated the optimal number of populations at 2. And then we made a tree using splits tree, which is a sort of a network type analysis. And so what you've got here, here's the wild group, here's the cultivars group, and then here's the mixed group, which appears to be very much an admixed group between the cultivars and the wild group. So our interpretation right now is that the mixed group probably represents hybridization or introgression or something involving the cultivars plus wild clade and the wild group. And my inference is that this represents contamination of wild populations from shiitake farms, which of course have incredibly high density of fruiting bodies and produce massive amounts of spores which go into the air. So this is of conservation relevance for sure. One last thing about ITS, when we look at the individual reads for the ITS in, say, the Shao genomes, what we find is that there's a lot of polymorphism. So we're just starting to work up these data now, but it's pretty clear to us that there's intragenomic heterogeneity in ITS in some of these things that have appear to have group 5 ITS. So we interpret that as probably some kind of ancient polymorphism that is just sorting out in unpredictable ways. Okay, so phylogenomics, the conclusions here are that the genome data really do largely support the ITS-TEF1 phylogeny with some exceptions. One is that Aciculospora, the thing from Costa Rica, looks like it's the sister group of all Asian Australasian things. ITS group 1 and group 5 are not supported as independent lineages, so limits to ITS-based species limitation. Edodes sensulato contains at least 2 lineages that I think might warrant recognition as species, which correspond to this wild and cultivars plus wild group. The mixed group may be some kind of hybrid or introgressed population. More to come on that later. And then there's intragenomic heterogeneity in ITS that might reflect some kind of maintained polymorphism, but we're still figuring that out. Okay, so finally, I want to tell you about some outstanding mysteries, and I apologize, I have to throw you a curve and tell you about Lentinula in Africa, which I have sort of misled you up to this point by suggesting that Lentinula is only in Asia, Australasia, and the Americas, and I apologize. I hope I haven't violated your trust. So you'll recall that we thought there were 13 lineages of Lentinula that may warrant recognition as species, 5 in Asia, Australasia, 8 in the Americas, or so we thought. A few years ago, actually quite a few years ago, Bart Buyck, who's in Paris, in the course of a survey of edible mushrooms in Madagascar, found a fungus that he said was a Shiitake lookalike. So he thought it was Lentinula edodes or something very similar. I overlooked this paper for many years, found it, freaked out, asked Bart for samples. He sent me some very tiny pieces of the herbarium specimens. And my postdoc, Brian, got large subunit data, which very strongly placed it within this Madagascar thing within Lentinula, within the Omphalotaceae, which is the broader family in Agaricales. He also then got ITS and TEF1, which suggests that it is, here it is up here, we're calling it Lentinula madagascarensis, which based on these data anyway, come out as a sister group of Aciculospora in Costa Rica and Ecuador. And so we've just published this in the journal Fuse, Fungal Systematics and Evolution. This was in the title slide for the whole talk. You might have looked at this and thought it was Shiitake because it really kind of is a Shiitake lookalike. So at least one Lentinula in what is at least politically Africa. But it's not the, and if so, that's a 4,000-mile range extension for the genus. But this is, turns out, probably is not the only Lentinula in Africa, because if you go to the website of the Meise Botanic Garden in Belgium, here's something that they report from the Democratic Republic of the Congo. They're still calling it Lentinula edodes, but we'll see. And we're in contact with them about doing some work together. And it does not look like Madagascarensis. So I think it's a different species in Africa. So I would bet that these two are going to turn out to be closely related. But even if they are, we probably have to invoke long distance dispersal to get Lentinula to Madagascar, because I believe Madagascar has been separated from the African continent for something like 150 million years, much older than the genus based on our molecular clock analyses. Other mysteries, something called Lentinula platinidodes has been reported from Vietnam. This is the data are not available. This is large subunit. We don't know, but it may be another lineage in Vietnam. And in this tree, at least it comes out close to the things in the Americas. So interesting. And then there is the most mysterious of all, Lentinula huarapiensis, described by Spegazzini. Here's a picture of one of the Exicati specimens. Doesn't look like a Lentinula to me, but who am I to question the great Pegler? So we'll see. Someday maybe we can get some DNA out of this. Okay, so I think a general message here from this work, well, there are a couple general messages. One is that international collaboration is really fun and rewarding and can be incredibly productive. So I want to thank all of my collaborators and everybody who supplied material to this project. And the other general message is that even in one of the world's best known genera of mushrooms, there are still a lot of mysteries, still a lot of surprises. The diversity of Lentinula in Africa is probably greater, I would guess, than just these two things. Perhaps one of you in the audience will find the next species of Lentinula in Africa. And with that, I will stop and I'll be happy to take any questions.
Thank you very much for a wonderful talk, David, particularly going from, I guess, the taxonomy and morphology through to certainly where we've been, one gene, a couple of genes, and then, wow, you have the whole genomes and some of what actually can be done.
Yeah.
I have a couple of questions. But I do see that Nicola has put in the chat line a question that I thought I would start with. And what she's saying is, with regards to the potential hybrid that you have in that Chinese-Japanese grouping, the intrabreast mixed group, could you generate artificial hybrids to examine their genomes? I guess it's going back to the fact that when you were talking about the earlier work that you were doing in Japan, that it was known that you could actually cross these different groups?
Yeah, absolutely. Before I answer that question, let me just comment back on your comment. Brenda, you're right. I mean, we have been so fortunate. I mean, when I say we, I mean you and I and many others, that we've really lived through two major revolutions in systematics, right? The initial molecular revolution and now the genomic revolution. And at every step, just a quantitative change in how we understand our things. So yeah, it's been great. Yeah, that's a really good question from Nicola. I guess what I would say is I think that the artificial hybrids have already been produced, and that's what's in this mixed group. So that's my inference that is what they are, that they are hybrids or something. Certainly one could do crossing in the lab and make hybrids of anything in Asia, Australasia, except this group 4 from New Guinea, right? So group 4 from New Guinea is reproductively isolated from all, or at least partly reproductively isolated from everything else. But the rest they mate. And I mean, I think that just really illustrates the great weakness in the sort of mating criterion for species delimitation. The Asian Australasian group is fairly young, and those populations have retained this plesiomorphic ability to mate. So that's not really, in my book, a good criterion for recognizing lineages. Yeah, you could make all kinds of interesting crosses and then do genomics and see, and do functional genomics and see what happens next. That's wide open.
So in actual fact, you've answered the question. One of the questions that I wanted to ask was your take on using crossing, the ability to mate with regards to delineating species. So I'm not going to ask that question. I could see that Tuan would like to ask a question, so I'm going to pass over to Tuan.
Thank you, Brenda, and thank you very much, David, for a very interesting talk. So I have a question connected to that hybrid mixed group in the middle. So if we recognize the first group and the bottom group, the white and the white cultivated group as species, how should we treat the hybrid? Because from what I see is that they actually seem to be, there's a coherent lineage, there's a diversity in them, and they also make their own trait. So are they become a species? Should we treat them as a species, or should we treat them as a hybrid entity?
Yeah. Yeah, nature is frustrating sometimes because we have these nice categories that we would like to put things into, and then things turn out to be messy. I mean, right now, my interpretation is that these look like, I mean, it looks like they are hybrids of two independent lineages. That's my interpretation right now. We're doing some phylogenetic network analyses to try to see if we can detect introgression and directionality, and preliminary results suggest that the scenario I described in words is what is happening. If you're asking about taxonomy, I'm not sure exactly what is the optimal way to go, but I tend to think that your species level taxonomy often reflects what you know and how much you care. So, I mean, we care a lot about Lentinula idodes sensulato because it's economically important and so on. So I would be in favor of formalizing those, one of the wild group as a new species, because I think that would potentially raise awareness about genetic diversity and the risk to the native populations that these shiitake farms pose. So yeah, just decisions about species delimitation, I think those are some of the hardest in science. And I think that they have a, I don't know, a political dimension or a practical dimension. This is a really important group of cultivated mushrooms, and we could communicate our understanding of that genetic diversity to the rest of humanity most effectively, I think, if we just gave it a new species name, because most like shiitake farmers and conservation people and so on, they don't want to talk about coalescence and introgression and stuff like that. But if you say, this is a different species, they get that. So I would favor that based on what we know now. But as far as those hybrids, should they be taxonomically recognized? I'm not sure. I'm not sure. And I'm still not 100% certain that they are, in fact, hybrids and not some kind of polymorphic population that's just retained some ancestral diversity. I think that's less likely. But I'll tell you, I'm much, for me, I'm a systematist. I'm most comfortable at the level of gene trees and big phylogeny. So when you get down to population level stuff and introgression and all that, it's challenging for me.
Thank you.
Yeah, certainly the one question I have is, within the group that includes the cultivated species, do you have any evidence that you've got some gene flow from that other species?
Yeah. So as I was mentioning just a minute ago, we have, well, first of all, we have that the SNP-based admixture analyses, which suggest that there are, if k equals 2, that's the optimal number of populations with some admixture between them. And we're starting to do these network analyses, which make it look like there has been some introgression from the wild plus cultivated group to the wild group. One of the problems with any Edodes that you collect in the wild in Northeast Asia in particular could easily be an escaped cultivar. So just because you found something in the wild doesn't mean that it's indigenous. Yeah.
No, it's fascinating. I mean, and certainly what does come to mind is the example of, it's the silkworm, bombyx, is suggested that it's no longer alive in the wild, and can one say the same of Shiitake, or the cultivated one? It seems like it is very much alive in the wild. So maybe the selection for cultivation hasn't been quite as strong to remove it from the wild.
Yeah, so something we'd really like to get into is look at domestication, see if we can see some genetic signatures of domestication. Because my read on Shiitake, which is not very well informed, is that it's sort of semi-domesticated. It's really not that far from the wild strains. So it's not like it's not like wine yeast or cows or something like that, or wheat. Yeah.
Don't even get me started on wine yeast as a domestic organism. You were talking about where you started your PhD. That's where I started my PhD, was on wine yeasts. So I see Nicola. In fact, maybe I should just mention to the audience here, and I've kind of stopped counting. I think we got up to, was it 120? Sorry, I'm looking at Neriman here. We got up to 120 people who were listening online. So I've lost track a little bit of who's online with us. And Faunus has suggested that if people have questions, they can put up their hands, but it's quite difficult to check the hands. But it's easier if people actually write things in the chat box. And Nicola has also written, "Thanks for the great talk, David. I agree completely about the difficulty and political nature of species recognition. And I work on groups that have much less economic importance, but perhaps in angiosperms, we can have a lot more morphological characteristics that we can use." So
yeah, taxonomy, if I can just editorialize for a second, taxonomy is definitely political. And I think we systematists need to kind of own that and just be aware of that we are part of the real world, and how you name things matters. I think there's a very interesting discussion that's come up recently about the use of indigenous names in taxonomy. One of the things that kind of bothers me about Lentinula edodes is that it was named by the Reverend M.J. Berkeley based on material that was collected from a shop in Japan. And it's been known as Shiitake or Shangu or Pyogo for many centuries in Asia. And so why should it be edodes, when there were pre-existing names used by the people who live where the thing comes from? So that's an interesting discussion that's starting to happen in systematics right now. Yeah.
I was going to say, this sounds like this would go completely against the code. What you're almost suggesting is a renaming of things. I mean, this.
It would be another, it could be incorporated in the code. It would just have to be explicit as a valid criterion for conservation, you know, and given the conservative nature of the botanical nomenclature community, I don't see that happening in our lifetimes, but maybe I'm wrong.
It's an interesting idea. I'm though a bit mindful of time at the moment, and I'm seeing another comment from Carlos Rodas, who's actually logged on from Colombia, and Carlos is saying thank you very much for a wonderful presentation, and he's wondering what the population in Colombia looks like for this interesting group. And from what you're saying, I don't know, do you have Colombian isolates?
I don't think I have any. Do we have Colombian isolates? I don't think we do. Thank you, Carlos, for the question. I have been, I mean, there's a really strong group of there's a really strong fungal taxonomy community in South America. My primary collaborators are in Brazil, Noemia and Nelson. But I've been talking to some folks in Colombia as well. Lentinula certainly is in Colombia. Raphanica is there for sure. Aciculospora may be there. I think that's a place, I mean, throughout South America, where just more field work and basic taxonomy is really needed. But Lentinula is most assuredly in Colombia. I've seen lots of pictures. You know, these days people put pictures on the web on all kinds of websites, Mushroom Observer and so on. So for sure it's there, and a field survey in South America would be great.
Well, I'm sure Carlos would love to host you in Colombia. Carlos spends a lot of time in plantations, and I'm sure sees a lot of very interesting mushrooms. But sorry, Dave.
No, just that they're typically in the tropics. They're typically at elevation, usually on various angiosperms. But I'd be happy to talk further later.
But I think we can. I'm now speaking for you, David, but I'm sure you'd be very interested in engaging with anybody in this group if they have any further questions, or want to send you some pictures, or even mushrooms. I'm sure that you would be very happy.
I'd be really happy, and I mean, Africa. I mean, there's got to be a lot more Lentinula in Africa. I mean, I don't know when it's going to. I don't know how easy it is to go to the Democratic Republic of the Congo for field work these days, but that would be great.
I would say that it's not exactly a walk in the park. So do I hand over to Merriman now?
Franz, we can also hand
Okay, Franz, we're going to hand over to you.
We're going to ask Bernard just to do the final word of thanks for David, and then we will continue.
Right. Franz, you want me to do it now?
Yeah.
Yes.
All right, sure. David, yeah, what a pleasure to listen to you, to sort of walk this historical route with you, and also seeing how the systematics of this group and reflecting on how that's changed for mycology in general, but also more broadly. And as you say, what an exciting, new time of discovery and understanding lies ahead for us. And you've raised so many tantalizing and interesting questions, everything from the naming to the diversity, and speaking to all of our desires to get out in the field in many different places of the world and collect. And we look forward to doing some of that with you somewhere in the future. Thank you so much for a fantastic talk, stimulating many thoughts and ideas for us. We look forward to continuing this discussion with you, and to see you again soon. And until then, I hope you stay safe.
Okay, thank you, Bernard, and everybody. It's been really fun, and I think I'm sticking around for a couple more presentations, right?
Yes. So from the society side, I also would like to thank you for being our international invited speaker. And now we will move over to our two local presentations, and Wilma Nel would be the first one. She is a PhD candidate, but I've seen today that they are already organizing a prestige seminar, which tells me that her reviews came in and that she would soon be Dr. Wilma Nel. But over to you, Wilma.
Thank you, Faunus. Can everybody hear me and see my screen fine?
Yes, we can, Wilma.
Okay, thank you. So hello, everyone, and thank you for still being online with us today. And thank you to Faunus and the Society for allowing me to present. And today I am going to tell you a story about some Ophiostomatales fungi we found in a very interesting place. So my story starts with a collaboration that was formed between FABI and researchers from the University of Wageningen in the Netherlands and the University of Copenhagen from Denmark. And this collaboration was led by Professors Dur Arnen and Michael Paulsen. And it was aimed at looking at the microbial diversity associated with fungus-growing termites and their mounts. Now, the fungus-growing termites are one of 3 insect groups that are considered true fungus farmers, as they cultivate and maintain fungal crops within their nests. And these fungi that they cultivate belong to the Physiological genus Timitomyces. And they maintain this fungi as combs in the subterranean parts of their nests. To ensure nothing happens to their food source, the termites actively tend the combs to keep them free of parasitic microbes. But as soon as you remove the termites, parasites do move into the system, and we often find other fungi cultivating these combs. And what was interesting to us was that after a few months of removing the termites from the comb and them not having to tend the Timitomyces, ophiostomatoid structures started to appear. Some of you might be familiar with one of the more famous species of ophiostomatoid fungi, Ophiostoma umi, which is responsible for Dutch elm disease, and it's vectored by the small bark beetle between hosts. Like Ophiostoma umi, many ophiostomatoid fungi are primarily found on woody hosts, and they are vectored by bark and ambrosia beetles, as well as the mites that they vector. However, we've been finding some species in more unique places. So we've found them in Protea and fructicenses. There are human opportunistic pathogens and even some nematode-associated species, but none so far have been discovered in any way associated with termites. Unfortunately, throughout the years since their discovery, there has been a long history of taxonomic confusion within the ophiostomatoid fungi. And we use the term ophiostomatoid to collectively refer to 2 different families, the Ophiostomataceae and the Sororicistidaceae, because these 2 families produce very similar sexual and asexual structures. During the time of morphological species description, one of the only ways to distinguish between species from these different families was based on their sensitivity to the fungicide cyclohexamide. And so species of Ophiostomataceae were found insensitive to cyclohexamide and could grow in its presence. And species from the Sororicistidaceae were sensitive to cyclohexamide and cannot grow in its presence. But since DNA sequencing was introduced, it became much easier to differentiate between the species in the respective families. And it was shown that these families are not closely related, but reside in different orders of the Pseudomycetes, and that the similarities in their structures is likely a result of convergent evolution due to the similar types of habitats that they share. But most of these species that we find in more interesting locations reside in the Ophiostomataceae family and the Ophiostomatales order. And so we wondered if these structures on the termite fungus combs belong to new Ophiostomatalian species. And to determine if this were the case, we followed a very conventional approach. I picked single spore drops from the sexual structures and transferred them onto Ophiostomatales specific media containing cyclohexamide. And then from the colonies that grew on this media, I transferred hyphal tips until I obtained pure cultures. Then I extracted DNA, did PCR and sequencing and phylogenetic analysis for 3 different gene regions, and I also looked at the morphology of the fungi. Now, following this conventional phylogenetic approach, I ran into some conflicting results. In the LSU phylogeny, the isolates separated into 2 distinct lineages, and both these lineages grouped close to the genus Ophiostoma. So for the ITS and beta tubulin data, we focused our analysis around Ophiostoma as well. But in these phylogenies, instead of grouping within the genus, our isolates fell basal to the genus. Additionally, the isolates representing lineage B that grouped as a single lineage in the LSU phylogeny separated into close sister lineages in both of these analyses. And we also found that for the beta tubulin gene region, it had different exon-intron compositions. So for lineage B1, it only contained intron 5, and for lineage B2, it contained intron 3 and 5. So based on these single gene phylogenies, we were quite confident in the amount of species we were working with, but we weren't sure about their higher level taxonomic placement in the family. So we decided to do additional gene sequencing, and we included the elongation factor 1 alpha gene. We did sequencing for the RPB2 region, and then we also did family level phylogenetic analysis for the ITS data that we had generated. And in each of these different phylogenies are isolates grouped at different locations. So for elongation factor 1 alpha, again, the isolates grouped closer to species of Ophiostoma. For RPB2, they grouped close to species of Sporifex. And for the ITS, they grouped basal to all the genera in the family. So because these conventional methods were helping us to resolve the higher level taxonomic placement, we instead decided to take a genomics approach. So we selected 1 isolate from each lineage A and B and extracted their DNA and sent this off for sequencing. And after assembling the genome data, we found something very interesting for lineage A. So where the genomes of species from the Ophiostomatales so far sequenced range between 19.5 megabases and around 44 megabases, the genome for isolate A, or the isolate from lineage A, was only 16 megabases in size, which is quite a bit smaller. So we just double checked the completeness of the genomes using BUSCO, and they were fairly complete, so we were happy moving forward with them. And at this moment, we have genome sequences for species from 11 of the 12 described genera in the Ophiostomatales, so we could take a phylogenomic approach to determine their higher level classification. And we chose 2 methods of phylogenomic analysis. The first approach was super matrix analysis, and in this type of analysis, one extracts all the core shared genes between the different species genomes, and then use these to perform a genome scale multi-locus sequence analysis. So we take those core shared genes, concatenate them into one large supermatrix data set, and then we moved on with maximum likelihood analysis. The second approach we followed was super tree analysis. And in this case, instead of concatenating all the individual data sets, one performs phylogenetic analysis for each data set, and then you superimpose all the different topologies of the individual gene phylogenies and extract the most accurate representative phylogeny from that. So we used 3 different applications of these methods. For the supermatrix, we performed maximum likelihood analysis using RAXML, and we used 2 different super tree algorithms. The first was STAG and the second was Astral. We just did the different methods to compare if there were large differences in topologies generated when using the different phylogenomics approaches. There were a few differences between the phylogenies, but overall they were quite similar. And in all cases, both our lineages grouped within the Ophiostomatales family, close to Ophiostoma, but they grouped as distinct generic lineages. So we were confident to describe them as new genera. I then also looked at the morphology for the 3 species lineages. For lineage A, like its genome, its morphology was also quite unique. And In this case, it produces golden brown sexual structures, where other Ophiostomatales species typically produce dark brown to black sexual structures. And these structures also maintain the flexibility in their necks throughout their lives, whereas with most Ophiostomatales, they become rigid and they break quite easily. And then for the two sister species for lineage B, we could confirm morphologically what we saw in the phylogenies as well, with lineage B1 producing a single asexual state with ovovoid conidia, and lineage B produces both this sexual state, or asexual state, sorry, as well as a second asexual stage characterized by round conidia. And based on all of these results, we then introduced two new genomes, Chrysopheria and Intubia, and we introduced the species Chrysopheria yanellii to represent lineage A, Intubia macrotomitonerum to represent lineage B1, and Intubia urlamansii to represent lineage B2. And so to conclude my story, new and interesting species can be found in some of the most unexpected of places as long as you look for them. And currently, morphology and DNA sequences are still a very powerful tool in taxonomy, but when it comes to the higher level classification of some genera, these tools alone might no longer be powerful enough to provide clear answers. But with more genomes becoming available, phylogenomic analysis can become a powerful tool in addressing questions like these. And without our phylogenomic analysis, we would likely have incorrectly described these species in the genus Ophiostoma. Lastly, I would just like to acknowledge all of the different funding bodies, Professors Drew Arnon and Michael Paulson, Professor Brenda Wingfield, who supplied the funding for the genome sequencing, and Mrs. Miranda Proctor, who shared her data during all those different phylogenetic analyses. Thank you very much.
Thank you very much, Wilma. I think we have a bit of time just to ask one or two questions. We will afterwards also allow people to ask questions. So please, if you have any questions, there's nothing I've seen in the chat line, but if you want to, just raise your hand.
Maybe can I ask a question?
Yes, Brenda.
Everyone's got good control. Who's controlling the camera here? This is excellent. Wilma, thank you for a really lovely presentation. And I mean, you served to focus my mind maybe a little bit more on these fungi. What do you think about the fact that that one genome is so much smaller? If I remember rightly, that's the one that has the funny beta tubulin sequence as well.
Yes, it does. So initially when we found these fungi, because they're occurring basically on the body of a different fungus, we considered that they might be mycoparasitic. And so we did a few tests with them. And so for the two species in Intubia, we didn't really find any results. But for this one species with the small genome, it does appear to derive some benefit from the Tomatomyces as well. So it might be actually gaining something from growing on the other fungus. And because of that, it might also have led to a reduction in its genome because it doesn't have to produce all of its own compounds because it's getting it from the nutrients that it's breaking down.
Yeah,
if that makes sense.
Really interesting. Thank you.
Any other questions that you would like to ask, Wilma? Well, you still have time to think about that because we, yes, David?
Yeah, just really cool talk. And I was just wondering if you had thought about looking into like the CASI profiles or anything, get any clues on the functional biology of these things? I mean, the phylogeny aside, and I mean, any ideas about how they make a living?
So I've looked at a few different protein coding genes. So I've looked at the Kazon profiles and the secondary metabolites, but they do look relatively similar to some of the other Ophiostomatales that I've compared them to. But I think I do need to go deeper and figure out what's going on because something's interesting there.
Okay, there's another question from Nicola. and she's asking, thinking of ulmi, which is a potential tree pathogen, are there any indications that these fungi could be found outside of the termite nests, especially on living plant material?
So I guess it could be possible, but there's no really closely related species to these ones that we found. So it might be something to look into in future, but at this stage, we haven't found them anywhere else except in the termite mounds.
Okay, Mike, you wanted to ask a question.
I was just telling you, Forrest, that there were questions in the chat box.
Okay.
I have a second, and I know you want to move on to the next talk. And David, again, thanks for a fabulous talk. So Wilma, yeah, there must be more of these. There are many termites, many species of termites. What do you think about that?
I think we need to dig up some more termite mounds and go find them.
Yeah, I mean, there must be many. How do they get? They're not being carried by the termites, so how do they get there?
Yeah, so we've thought about it being on the substrate that the termites carry in, but if you look at the process of these fungus growing termites, they don't just take the material into their mounds, they actually eat the material and then digest it and then deposit it into the termite mounds for the Timothymices. So if the termites are taking it into their nest, it actually has to survive the guts of the termite, which would be quite amazing as well.
Or there are mites that are associating with the termites or with the rest of it.
Yes, but it's definitely interesting to go and explore.
Well, that's the beauty of science, that there's always a lot more to do. And I always say that the one thing about us is we can never be, you can be wrong. You can say, sorry, I made a mistake. And the other thing is there's always something new. So those are the two joys of being a scientist. But let's move on. And Aiden, we would like to hear your talk now. As I say, afterwards, we will also allow some time for questions. And thanks to Wilma for a very nice talk.
Okay. Can everyone hear me?
Yes, we can.
Okay, thank you very much. Good afternoon, everyone. My name is Aiden Visagie, and I'm going to be presenting to you today about my master's work, which had to do with the isolation and identification of Penicillium from wheat-associated agricultural soils. Penicillium is a genus of Ascomycetes fungi which occurs worldwide in a variety of different environments. This genus is typically characterized by their brush-like conidiophore structures and exhibit a large rate of species discovery, with over 120 new species being discovered between 2014 and 2020. Penicillium play an important ecological role as saprophytes and as nutrient sources for soil-living organisms such as mites and nematodes. They play an important medical role as sources of new antibiotics such as penicillin, which is the most well-known example. They play an important agricultural role, both as post-harvest pathogens such as P. digitatum and P. expansum, and as phosphate solubilizing or plant growth promoting microbes such as P. baeyiae. South Africa is also a known biodiversity hotspot for Penicillium, particularly the Cape Floristic region or the Fynbos biome, with several new species having been described in this biome, and the most recent that was also isolated in this project was Penicillium alsopiae, and it's mentioned specifically because a very large amount of it was isolated in this study, and we used it for a potting trial later in the study. So the methods of identification in Penicillium are morphological, physiological, and phylogenetic in nature, and a combination of these methods is used when describing new species, as the currently accepted species concept in Penicillium is a polyphasic species concept. So when looking at morphological classification, I'm going to just show you guys pictures of a novel species which I found during this work, which belongs to section Rolfsiorum, a series Rolfsiorum of section Lanata divaricata. As you can see, the morphology changes drastically depending on the media that you put it on. So we look at a variety of different media when we're noting morphological characteristics, and you note all visible morphological characteristics. We also look at thermotolerance in this genus, as it can be very taxonomically informative because the temperature has a very large influence on the growth rate and the morphology of these fungi. When we do micromorphological classification, it involves the examination of the conidiophores, or the asexual reproductive structures. These pictures to the right are pictures of my novel species, which I found, and its notable features are a lack of metulae and a lack of a subterminal branch. It is also sclerotogenic, and the reason why the subterminal branch is noteworthy is because that is what distinguishes it from its closest phylogenetic relative. So phylogenetic analyses in Penicillium is done primarily using 4 genes: ITS, beta-tubulin, RBB2, and comodulin. ITS has weak resolution within certain sections of these genes, so may not necessarily resolve to species level. So therefore, we chose beta-tubulin as the primary marker. And when the species was in doubt, or when a new species was being described, we did the additional gene sequences to perform multi-gene phylogeny for GCPSR, or genealogical concordance, the phylogenetic species recognition. These are just examples of this comodulin tree, RPB2 tree, and a Ben A tree, and then the concatenated tree where P. alsopiae was described, which is the novel species that I used for my potting trial, as we saw a very large amount of the species in wheat growing fields, which is strange considering that it is supposed to be a native fynbos strain or species. And this is my concatenated phylogenetic tree, which shows that my strain, which is morphologically distinct from P. pulvelorum, also with very high phylogenies, is phylogenetically distinct from P. pulvelorum. The physiological analyses I did not get to do in my study, but how it is performed is via growing up the fungal strains of Penicillium on CYA and YES media, after which extralytes are extracted from these strains, and then a HPLC profile is generated using this extraction. These HPLC profiles are species specific, so it can be very useful for taxonomic classification. In this study, we focus on agricultural soil environments from 3 different farms, which differ mainly in either crop rotation or soil composition, the sampling sites. The classification in the study, as I mentioned before, used mainly beta-tubulin, unless looking at potentially novel species, and morphological classifications were done to provide the additional data to verify the phylogenetic classifications. Plant growth promotion activity was also examined, so we looked at indole production and phosphate solubilization activity on plates, and we noted very weak activity of the indole acetic acid production plates and very high phosphate solubilizing plates, phosphate solubilizing activity. So we chose, based on the phosphate solubilizing activity, 5 organisms for a potting trial. They belong to the 5 species as listed in front of you: P. alsopiae, P. corvianum, P. mercianum, P. bellaeae, and P. pilonicum. The potting trials were performed by applying 3 concentrations of fertilizer at a full commercial dose, half commercial dose, and quarter commercial dose, and the negative control and treatments were supplemented with inorganic phosphate analogous to the amount of fertilizer applied to the positive control. The organisms used for the potting trials were grown on a modified PVK medium, as this induces phosphatase activity. We made spore suspensions, and then we standardized these spore suspensions and applied this organism at a level of 5.7 times 10 to the power of 7 spores per milliliter to the seeds of the wheat seeds, after which the wheat seeds were planted and left for 3 months. And so just going to give you guys a quick overview of the diversity results and the potting trial results. So what we noted is that with the wheat crop rotations, there was a far higher instance of Aspergillus, and there was far fewer, there was a net reduction in the amount of fungus which were isolated. The wheat medics also contained Penicillium alsopiae and Penicillium nov, so it had both the novel species, but the wheat did not, so it indicates that the monoculture is selecting against these novel species. The wheat canola was by far the most diverse, and yeah. For the 3 farms, we noticed that Taexelruk was by far the most diverse, and followed closely by Langewens. The notable difference being that Taexelruk had a far higher number of fungal isolates, as it was more nutrient rich. And Hopefield was by far the most nutrient poor and the most well drained, so it had very, very low amounts of diversity and low amounts of fungal isolates. In the potting trials, we saw there was no significant difference for the shoot length between the species, and a very small effect seen for the plant yield of the species. And of all of the treatments, the initial fungal strain was successfully re-isolated except for treatment 5. So P. Phylogenicum strain, which was applied, did not survive till the harvest. We saw significant differences in root weight and volume between the negative control and treatments 1 through 4, whereas 5 had no difference. And the positive control, interestingly, showed no significant difference in root weight or volume when examining the lowest and the highest dosages, but it did at the middling intermediate dosage of inorganic phosphate. And one interesting thing to note is if you look at 1H1, the circles there, it shows that although the root weight of the first treatment at the higher inorganic phosphate dosage did not massively increase, or for treatment 3, the root weight actually decreased, there was a very large increase in volume, which means that at this higher dosage of phosphate, there was a far higher degree of root branching. So there's far more root volume being formed, indicating that this fungus was having some form of positive association with the wheat plants. So in conclusion, crop rotations and farming practice impact Penicillium diversity. Novel species of Penicillium are likely to be found from many habitats, including agricultural, which we don't necessarily think of as a diverse environment. And some of the native Fynbos strains isolated show the ability to influence plant root growth, particularly wheat root growth, which is interesting because it means that the wheat successfully associated with what is supposed to be a native Fynbos strain. And large numbers of this native Fynbos strain were isolated in this study from all 3 different crop rotations. So it does appear as if it is successfully associating with commercial crops in some way. I'd like to thank the following people. And are there any questions?
Thank you very much, Aiden. I forgot to say that Aiden is a MSc student at the University of Stellenbosch. And thank you very much for being willing to present today. And I see there are some clapping of hands because there's no real audience that can do anything. So Nicola has got a question. Thanks for your talk. I learned a lot. In particular, I had not known that the CFR had high Penicillium diversity. I especially enjoyed the beautiful images of your new species. So she's giving you a compliment. Any person that would like to ask a question to Aiden? You can just indicate. Sometimes it's easier to speak than to Brenda.
So, I mean, we can't let Aiden off the hook here without any questions. So, Aiden, you're a master's student and you described for the first time a new species. How do you feel about that?
It's strange because it's so interesting to me that no one's seen it before. You know, I think that was quite a cool moment sitting in front of the microscope and looking at it and taking the image and realizing like, I'm the first person to have seen this. So it's a wonderful experience. I think it really made me, it drew me more into the field than what I was when I started, because I was a bit unsure of if I wanted to do mycology, but now I'm quite certain I want to continue with it. So I think it's
so you're hooked, totally not onto penicillium. I mean, that's an awful fungus.
Hopefully, yeah, we'll see how it goes.
Corvus and I joke.
Yeah.
Corvus is crying about that.
He disagrees with me.
Any other questions that you would like to ask Aiden? And if you had some time also to Wilma? Nobody at the moment?
I can maybe...
Yes, Corvus.
Excuse if Luna makes a noise in the background.
That's fine.
Aiden, considering that the farms that you're collecting from sits in the Fynbos area, don't you think that your soil will be more diverse than what you would usually find in agricultural soil?
Yes, so that is something that I wish I had included as an additional sampling group, is a commercial agricultural farm outside of the Fynbos region as a comparison or as a negative control almost. because I definitely think that, especially when you consider the fact that a lot of these penicilliums are air dispersed, so it's not too outlandish to think that they would travel just 2 fields from the native wild fynbos to the farm, because the farms aren't very far away from native fynbos. Yeah, so I think that's definitely plausible.
And just to follow up that, on the phosphatases and growth studies that you're doing, I really think that the next step for us in Penicillium is to take on the genomes. I know there's a big effort sequencing 1,000 Aspergillus genomes, and we shouldn't be left behind in Penicillium on this. So you should really think about sequencing some of those species that seems to induce growth in wheat, so that we can understand why it's doing that.
Yeah, I think especially given the phylogenomics talks of today, I've seen there's a huge amount of utility in having that data, because there's so many analyses that you can then perform afterwards, both in terms of the population metrics and in terms of what in there is usable to us or helpful to us.
Great. Any other questions? David has made a comment. He says, congratulations on a great talk. And he seconds the 1,000 Penicillium genomes project and says you should propose it to the JGI.
Okay. We'll set up some collaborators and then we see what happens.
Well, it's great because they're very unusual for a U.S. government facility, which is that, you know, that they will accept projects from anywhere. So the PI could be at FABI, for example, or Stellenbosch.
Well, that's wonderful. It's definitely giving me a lot to think about today. So I'm very grateful for this opportunity that I've been given by the Systematic Society to meet everyone here and present my work. So thank you very much for listening to me.
Great, and good luck with the final stages of your MSc. So any other questions that you would like to, Lauren James has a very interesting talk. It's great to hear that at least some indigenous species are surviving and even being discovered out of the wheat monocrop. So a comment there. Anything else that anybody that wants to raise a question, you're welcome to do so. While you're thinking about it, let me just do a bit of advertising. So as I said, the society, the Systematic Society, has got their seminars on the first day of the month, whereas FABI's is usually the last Thursday of the month. But this time they sneaked over into the 1st of July and we could make a joint effort. So on the 5th of August, we would have Dr. Sandra Knapp from the Natural History Museum in London, and she's going to talk about her work on the Solanacearum family. So potatoes, eggplant, all of those agricultural crops that we love to eat. So that's just a heads up. You will see the adverts and things coming out as we get closer to the date. Neriman, when is the next FABI International Seminar?
Just checking the calendar now.
29th July.
29th.
Yes.
Okay. So on the 29th, there will also be an international, and whether you belong to the one or the other group, we will inform you about these talks. And thanks a lot to everybody that stayed around and are willing to listen. I think it's a very, also thank you from FABI's side to the society for linking up with us, and then from the society to FABI for linking up with us. So I hope that you've enjoyed this, and there's still some scope for more talks. We're looking for young people to present their work in the society, so please remember that. The final part of, so let me ask, are there any other questions that you still want to raise to the speakers? Somebody saying in Kenya, we usually consider Penicillium a contaminant. Have you found a pathogenic species on plants?
So in the agricultural field, when I looked at it, I didn't see the pathogenic species, but it could also be that I didn't really look at that. And the pathogenic species in Penicillium don't often target wheat. So they're more often problematic for other crops, at least not pre-harvest. So I didn't look at any post-harvest environments. So in those environments, it would be far more likely to isolate those problematic strains.
Thank you. So what we can do now is say, yes, generally post-harvest, that's Quibus's comment. That's what Aiden said. So the last part of so again, thank you to Aiden, to Wilma, and to David for giving their time and coming to talk to us. I think we all agree that it was a wonderful seminar today, and thank you very much for making the effort and presenting very nice talks.